Hello, class 12th. In today's class, we will discuss about the development of an anther. In previous class, I taught you about the structure of an anther. In that class, we only focused about the structure of the anther, what layer is having the different layer having the different name. Today, we will see the development of those layers. Right? In the topic development of an anther. Okay. So the development of the anther starts on the thalamus, which is the basal part of flower. Okay. And in initial stage, the anther looks like it, having the two layer, the epidermis, which is the outermost layer, and the inside it, which you can see in the green color, is the outgrowth. This outgrowth is a group of many cells which is responsible for the further development. Okay. After this structure, you can see the different structure which develop from this structure. Okay, which develop from this structure. So here you can see the blue line which present outside of it is actually the epidermis, which you can see here also, right? And in each corner of the diagram you can see the four different cells which not looks like this green color. These are the different cells found in each corner of the anther. These are called the hypodermal cell. The epidermis is the outermost, and inside it you can see the hypodermal cell. These hypodermal cells develop and become the archisporial cell. And you can say the hypodermal cell is also called the archisporial cell. These archisporial cell is the mature stage of hypodermal cell. Right? When these become mature, these cells divide mitotically. And you know very well what happened in the mitosis. The parent cell divide into two new daughter cells. Right? Here you can see the two new cells. Actually, these are the cells those develop from the hypodermal cells or archisporial cells by mitosis cell division. Okay. So here you can see two new cells. The cell which present towards epidermis is called primary parietal cell. Okay. And the cell which present inside it is called the primary sporogenous cell. Right? So primary parietal cell develop the different lining of the anther, which I taught you in the previous class. These linings are the outer one, epidermis, inside the epidermis, the endothecium. Okay, inside the endothecium, 2 to 6 middle layers, and inside it, the tapetum. The different type of tapetum I taught you there. Okay, so these all layers develop from the primary parietal cell, or you can say the primary parietal cell is responsible for the development of all the layers of the anther. Okay. Inside it you can see, inside all the layers you can see this sporogenous cell, which is the primary sporogenous cell. Actually, it's develop and convert itself into the sporogenous tissue. The group of cell is called tissue, that's you know. So earlier it was the primary sporogenous cell, and after doing some multiplication by the division of mitosis, so now it is a group of sporogenous cell which is called the sporogenous tissue. Okay. Now after this you can see when in sporogenous tissue the development takes place. Now we will discuss about the development here, because layers development I already taught you there in the previous class. Okay, so here you just see this is the tapetum layer, which you can see the outer side is the middle layer, the outside of it the endothelium, and the outermost is epidermis. Okay, but till here the tapetum develop. Inside the tapetum in green color you can see the group of sporogenous tissue. Many sporogenous cells are here, and that's why here you have seen this is called the sporogenous tissue. Okay, so these cells develop, and after the development these cells take part into mitosis cell division. The continuous mitosis cell division develop the spore mother cells inside it. Right? Means the sporogenous cell, after doing mitosis cell division, the continuous mitosis cell division, these develop spore mother cells. Okay? These spore mother cells, now these spore mother cells will divide by meiosis process, the another cell division process. Okay, meiosis cell division, and that's you know very well. The speciality of meiosis cell division is what? By this cell division, the new cell formed, and these newly celled are haploid by nature. Those develop gametes after it. So here you can see the spore mother cells divide by meiosis process, and these will develop the microspores. These microspores are haploid. Okay, and after these will develop the pollen grain. So here you can see how systematically the processes takes place and develop the pollen grain from sporogenous cell. Okay, this sporogenous cell develop from primary sporogenous cell, and this primary sporogenous cell develop from hypodermal cell, which is also called the archisporal cells. Okay, after this process the more development takes place, and you can see here in this diagram these are the microspore cells which develop here. Okay, so these are the many microspore cells which develop by the cell division. Now look at here, around the microspore cells you can see the green color area. Okay, this is what? This is actually the carbohydrated liquid. This carbohydratic liquid is a nutritive liquid which provide the nutrition to the developing microspore cells. Right? And this nutritive liquid is called callose. So the callose provide the nutrition to the microspore, okay, in the time of development, and these microspore after getting the nutrition from the callose develop and convert into pollen grain. Here you can see after getting the nutrition the microspore cells become tetroid, and now these are called the tetroid microspores. These tetroid microspores are tetrahedral, having the tetrahedral arrangement. The three nucleus you can see here, and one is behind it. Okay, so that's why this is called the tetroid. After the development into the microspore tetroid, layers also develop, and the microspore tetroid divide and convert into many microspore here inside the pollen sac. Okay, and here you can see the complete structure of anther. Okay, the pollen sac, and inside the pollen sac many microspore or pollen grains are present. When these microspore will become mature and will convert into healthy pollen grain, the whole anther will dry up, and when this will dry up, the stomium portion, which is formed by loose parenchymatous tissue, that's I told you in the previous class. So this will become the weak area and will burst, and by that area the pollen grain will comes out and will go for the pollination by the help of different agencies. Okay, so it was the complete story of the development of an anther. Some biological terms are very important here, which I taught you. Focus these biological terms because these terms are very important while writing your answer in your exam, and from these terms the question comes into your competition exam also after the 12th. Okay, so this was the topic. Hope you got it, and if any difficulty will be there, so you can message me and I will give the answer of your question. Till that time, stay at home, save your life. God bless you, children.